Yeah, so this talk is on the molecular evolution of Inhibitor Cystine Knot toxins in a wandering spider, Ctenidae, and it's by Jeffrey Cole and Michael S. Brewer from the Eastern Carolina University. So take it away, Jeff.
All right, can you all see my screen and hear me?
Yeah.
All right, perfect. Yeah, so I just finished up my dissertation with Michael Brewer at East Carolina University. So I'm just going to be talking about some work from my dissertation. So let me do present mode. All right, so just a brief overview of Ctenidae family. So these are wandering spiders. There's like over 500 species, and 6 of those live in the US. And of those, they don't have venom that's dangerous to humans. But most of the diversity that's in this family lies within the neotropics. And so the wandering spiders that I mostly focus on are the ones that live in the US. And so there's Ctenidae, Ctenidae in Arkansas, Hibernalis in Alabama, and then Capiosis that's in Florida. And then in Texas, there's Leptotenus virus, which mostly lives in like the hill country in Texas. And then Anahita, which is like the smallest Ctenid in the US. So it kind of lives all throughout in the southeast. And what they're, we say wandering spider, a lot of people think of Brazilian wandering spider, so Phenutria, which is very different than the ones that live here in the US, at least in terms of behavior, because it does have much like nasty venom that's dangerous to humans and expresses a very potent toxin called TX1, which is actually a sodium ion channel inhibitory cystine knot neurotoxin that has a very low LD50 when you inject it into mice. And so there's been investigations in terms of like the transcriptome of this species and identified that there's lots of other inhibitor cystine knots, so we'll call them ICKS. Just in this one species, there were like 98 different toxins of 9 different like topological frameworks, and 6 of those were verified to be like inhibitor to conform to the inhibitor cystine knot fold. And so inhibitor cystine knot is basically it has these cysteines. They form these like disulfide bonds, and it makes like this knot-like structure, and they act as like neurotoxins to make this very stable neurotoxin, and it actually makes up the majority of a lot of different species of different spider species venom components. And so the loops between the cysteine residues can be very variable and sort of gives rise to different types of topological diversity, and also gives rise to lots of different specificity. And so think it can target invertebrates or vertebrates or different types of ion channels. And so as a recent paper investigated sort of like the evolution of inhibitor cystine knot toxins, and this was the sort of biggest investigation that sort of used structural data to find out that ICKs actually in spiders all share a common ancestor. And so I was able to use this framework as like an inspiration for my research, which was basically in wandering spiders, specifically like the Brazilian wandering spider, which has its really complex inhibitor or ICKs. Sort of what sort of how does that ICK composition kind of vary between other Ctenidae species, and so having such diverse ICK composition, what sort of selective pressures underlie the ICK diversification in Ctenids. And so I collected venom and venom glands from all of the US Ctenids that I mentioned before, and was also able to get some samples from Brazil, so Phenutria nigriventer, and then also another species of Isotenus. And then as well, I retrieved transcriptome data from the short read archive from different members of spider families that are in the RTA clade as like outgroups. And so for venom collection, I extracted out venom using electrical stimulation, dissected out the venom glands, do mass spec proteomics, and then sequencing the mRNA from the venom glands. And then once I had that data, that began my search for inhibitor cystine knots. So I developed this informatics pipeline to identify ICKs. And so basically just taking your genome or your transcriptome and your proteome and compiling those into peptide sequences and doing homology searches with BLAST and what's called HAMMER, and then taking in a data set of verified inhibitor cystine knots that have a complete open reading frame, and then filtering those further to find things that have a certain number of cysteines that are of a certain length, and then running those through signal P. So because all inhibitor cystine knots, these neurotoxins, they're secreted in the venom gland, so they should all have some sort of like signal peptide. And so that's just the sort of like overview of kind of the pipeline, and that gives out the putative ICKs. And then once I had the ICKs, it's a matter of trying to predict what the disulfide connections would be. And so this is actually a difficult thing to do informatically, because as you increase the number of cysteines, the number of possible disulfide bonds just explodes in a combinatorial fashion. So I used 4 different computational approaches to attempt to predict disulfide connectivity. And then once I had those disulfide connectivity predictions, I could use that to identify homologous cysteines, which then was used to guide a multiple sequence alignment of the ICKs of the inner cysteines, and then take that and then reconstruct the phylogeny. And then once we have that, doing phylogenetic tests for selection. And so the first one is just a very basic, just gene-wide. Is there any evidence of positive selection at all in the data set? And then you're looking for pervasive. So given the alignment, the codon alignment, basically, is there strong evidence are there any sites that are undergoing strong positive selection? And then looking at, instead of just sites, if there's within the phylogeny of the ICKs, are there any specific sites that may be undergoing positive selection? So that's just an overview of that. I won't go into too much details of that. If you have more questions about the positive selection, I can answer questions about that. And so this is just an overview of the different motifs that we identify for ICKs. So the simplest one had 6 cysteines, and then the most abundant one actually had 8 cysteines. And so basically, the X just means there's only one residue between it, and then a dash means that there's more than one residue between it. And so these are the different motifs that we found and how many we found of each. And so a total of 626 peptides were identified across these 27 species. And like I said, the most abundant one was this one right here that had 8 cysteines. All right, and so what this is a summary of the disulfide connectivity predictions. So this is for the 6-membered, the ICKs that only had 6 cysteines. And so you read this as cysteine 1, 2, 3, 4, 5, 6, and so on. And so this summarizes all of the predictions for the different approaches. And so a thick band indicates that most approaches said that those 2 cysteines would be connected. And then thinner band just means less things did. So for the 6-membered framework, it was pretty clear what the predicted disulfide connectivities would be. But as you added more and more cysteines, or pairs of cysteines, I should say, it becomes less and less clear, to the point that it's very difficult to actually infer what all of the disulfide connections would be. And so what we did to guide our alignment was just using the basically the three disulfide connections that we had the most confidence in that sort of shared amongst all of them. And so that's what's pictured here. So those are the three disulfide connections shared amongst all of the frameworks. And so we use that to build our phylogeny. And what we see is that the 8-membered framework is also the most abundant, but it actually appears that instead of it being an elaboration of the 6-membered one, it seems that the 6-membered one popped up a couple of times in the phylogeny, indicating that it's perhaps an elaboration of the 8-membered framework. And then we also interestingly found that this 12 cysteine framework is single copy, doesn't have any duplicates, and was unique to ctenids and their sister family, the Cychraids. So for the gene-wide positive selection, it did identify evidence of gene-wide episodic positive selection. But whenever we look at the specific sites in the ICK alignment, none of them had very strong, like pervasive evidence of positive selection. So what I've done here is just plotted the synonymous substitution rate against the non-synonymous substitution rate, and basically, if there were like strong evidence of positive selection, you would have things way up in here, but everything's indicative of very strong negative selection. In fact, about 57 out of the 80 sites had evidence of strong negative selection. But when we look at when we take the phylogeny into account and look for episodic episodes of positive selection, we find that there's 12 sites that have significant evidence of episodic positive selection. And so what's plotted here is the basically like the p-value or that it's undergoing positive selection. And so what's interesting in this is that none of the sites in between the second and third cysteines had strong episodes or strong signatures of positive selection, and it looks like it may be under or has evidence of negative selection. And so we don't know like I've showed you before, it's very hard to have a good idea of what the disulfide connections would be, and that gives you information about the structure. So we don't know too much about like the structure of all of these things, but this could perhaps give us some sort of insight into something about maybe the structure, in that perhaps this region here that's under seems to be under negative selection could be perhaps useful for like the core like toxicity, and then maybe these other regions could be like useful in like being more specific or honing in on different types of specific functions. And then other than that, instead of just looking at sites and then saying if a portion of branches have episodes of positive selection, looking at very specific branches on the phylogeny. And so you do that for like all of the thousands of branches in the phylogeny. And we found, and it only found 2 that had really strong evidence. And one of those branches was actually a clade of 19 peptides with the 14 cysteine framework, which was
You're muted, Jeff. Can you unmute yourself?
okay. Can you hear me
Sorry,
now?
that was me by accident.
Oh, okay. So you can hear me now. Okay, sorry. All right. So of the 19 peptides, one of them was a 14 membered or cysteine framework, and so it was unique to Tenus, so it excluded Tenus corniger, which was the only old world representative that we actually had. So all of the Tenus that were in that clade were new world. All right, and so the main thing to take away is, well first, disulfide predictions for spider ICKs, it's very difficult. There's lots of work to be done to make improvements in those regards. But yeah, often like the 6 cysteine framework, because it has less cysteine, so it's typically referred to as like the core ICK framework, at least in this analysis, appears to actually be derived from the more abundant 8 cysteine framework, perhaps through a loss of a pair of cysteines. And it's not too surprising that we don't find very strong pervasive evidence of positive selection, just because ICKs and spiders, they're hundreds of millions of years old. And so at that point, it's expected that negative selection would pervade. But it is interesting that we see episodic bouts of positive selection, which is sort of what, in terms of like understanding how venoms evolve and what how in terms of like the evolutionary arms race between like prey and predators, this could give us some sort of insights into how that could potentially go about. And so there's still more to be learned about the function and structure of all of these types of toxins. And there's more work to be done in terms of like the assaying their actual function, molecular modeling perhaps could be useful. And then also in terms of just understanding how these things have evolved. These are huge multi-copy gene families, which understanding like the actual patterns of duplication would require like whole genome sequencing. Right, and so thanks so much.
Yeah, we don't really have too much more time for questions, but there is one from Robert Hanley. How confident are you of codon homology between cysteine residues?
How confident am I in?
Oh, codon homology.
Yeah. So that, yeah, exactly. So that is something that I've spent many hours pondering over, and so the codon homology, it's based on just doing back translation. So I start with amino acid sequences, and then kind of like back translate to the codon. So the codon homology is based on what I have and the amino acids, and I really try to focus on things that are just within the cysteines in the analysis that I've done.